I want to tell you about this worm. It's 1 millimeter long. It has a gut, muscles, a neural network. And, I tell you about myself. I loved playing with worms and snakes as a kid, growing up in Revere, Mass., just up the road here. Revere was a kind of a tough, gritty town. And you would leave school and worry that you would get in a fight in the schoolyard. But I was precocious by local standards. I quit smoking in the 3rd grade. But back to the worm. We activated one gene in that animal, one, and it lived longer. Not only lived longer, it was healthier and more active right to the end. You have that gene. The problem is that gene loses activity as you get older. I've spent 30 years studying that gene, how it works, how it fails, and how to fix it. And that's the story for today. In the 8th grade, one of my teachers took my mother aside after school and said, "Pull him out of the public schools. They're not going to bring out the best in him." So the following year, I found myself at Boston College High School, Jesuits, rigorous, relentless. Just what a kid from Revere didn't need. But something changed, and slowly, I found myself a serious student for the first time. That teacher saw something in me and knew I just needed the right conditions to unlock it. 20 years later, I got tenure here at MIT, and I decided I really wanted to work on something new, something important, something risky. And I decided to study aging in my lab. Now, aging at that time was a scientific backwater. When I told my colleagues, their reactions ranged from disbelief to politeness, the kind you show when a friend shows up with a bad haircut. So we started studying aging in yeast cells. These are the cells that were the workhorse I rode to tenure in the first place. After about 6 or 7 years, we found that one gene in yeast, when we increased its activity, made yeast cells live longer. That gene is called SIR2. So SIR2 slows aging in yeast and promotes longevity. This was an era of genome sequencing, and the sequences of numerous organisms appeared, and all of them, amazingly, had one or more genes with a sequence similar to the yeast SIR2. These genes and proteins collectively are called sirtuins. The fact that they're so conserved through evolution says they must be important. So in a burst of optimism, I started a worm lab within my lab, and we focused on the worm's version of the SIR2 gene, and we activated it, and the worms lived longer, as I told you at the beginning. Now, this result was quite stunning, because what it really means is that sirtuins slow aging across organisms. And if it's true in yeast and in worms, it ought to be true broadly in nature and include mammals and include us. I was excited. I rushed to tell my department head about this new finding. And he said, Just what the world needs, long-lived worms. Evidently, the aging field still had some climbing to do. So the field grew, and we and other labs probed what these sirtuins actually do inside of cells. And we found that they repair damage to DNA, they repair damage to mitochondria, the powerhouses of cells, and they're master regulators of something called epigenetics. And epigenetics is involved in keeping the right genes on and the wrong genes off in all our different cell types to give them their identity. They're the cell's maintenance crew. So we were kind of dazzled at that point, but then we were hit with something even bigger, and it was a result that changed everything. We're studying these sirtuin proteins now, and they didn't do anything. They had no activity. And it turns out they're dead as a doornail without a compound called NAD. And NAD is found in all living cells. It's been assigned a few activities within these cells, but it's never been linked to aging before this finding. I told you at the beginning, you have this gene. And I told you that its activity declines as you get older. By the time you're in your 60s, the activity is reduced by about 50%. The reason it declines is because the levels of NAD itself declines as we get older through midlife. The sirtuin work crew still shows up early, but the fuel to run the machinery runs out by noon. This led to a big idea that NAD could be an intervention point in aging. If we replenish this lost NAD, we should slow aging. Around that time, I got a cold call from a young entrepreneur, and we went on to start a company in aging, and the first product was a pill to boost NAD. Now here's the thing, the pill itself contains no NAD. Turns out you can take NAD pills, you can take NAD intravenously to no avail because NAD is too big to get into cells. It just pokes around outside the cells, looking in. But smaller precursors of NAD, NR and NMN, do get in. And the cells have enzymes that add the missing part to create NAD. Just like adding the keystone to complete a sturdy arch. So we were ready to test the NAD pill, and we conducted a human trial in older adults, daily dose, double-blind, placebo-control, the works. And what we found is NAD rose from the low levels in these older adults to youthful levels. And they stayed there the duration of the trial. The pill works. Later trials showed health benefits followed. For example, in something called fatty liver disease and in the lung disease COPD. And a lot of other trials are in progress. And yes, it seems to slow aging as measured by following biomarkers. Millions of people now take NAD boosters. And like AI, it's still early days. So what about the future? Where do we go? I want you, scientists, engineers, doctors, builders, to hear this as an invitation. NAD is a platform, and platforms grow from their point of origin. to new places. Consider this: aging is the major risk factor for late onset diseases. So if you slow aging, you would simultaneously address many diseases all at once. Who knows someone with Alzheimer's? It's a lot of you. It's a scourge, for sure. Well, NAD decline in the brain is an early driver of brain aging. By replenishing lost NAD, can we delay or even treat cognitive decline and neurodegenerative diseases? Pilot studies, small studies, look promising, and larger trials are in progress now. Metabolic diseases. The NAD sirtuin axis promotes insulin sensitivity, fat metabolism, and lowers inflammation. Might boosting NAD become part of the normal treatment for diabetes? What about inflammation related to aging? And completely outside of the health box, what about space travel? A very timely subject right now. Future missions will feature vast distances and will spawn new space colonies. How can we keep the crews vital for missions that might take decades? Slowing aging might be one answer. Inflection points in life are rarely announced. They look like a cold call from an entrepreneur, an 8th grade teacher after class, a worm that refuses to die on schedule. Growing up, I was told, don't ask too many questions. This went in one ear and out the other. And ignoring the advice, I asked why yeast age, then worms, then mice, and had the chutzpah to say, what we found there would be true in humans. The scientific establishment thought I lost my way, but the worm didn't care about the scientific establishment. It just kept telling the truth. Look, we're not built to last forever. But at the same time, we don't have to submit to the fate of past generations. For the first time, we have the science to take action. That's why I'm standing here today. You know, someone in this room or who watches this video may take what we started to a place we can't even imagine. And if that person is you owe me your first $1 billion. Know this: aging is not written in stone. It is written in biology. And biology responds to the right question, asked at the right moment by the right people. You are the right people. This is the right moment. Find the right question, ask it, and stick with it.